Meiosis is an important form of cellular reproduction. It is used for sexual reproduction, that is, to make cells which are not identical to the parent cells. The cells produced by meiosis are sex cells, or gametes. These cells are used for reproduction. It is important to remember that in meiosis, the daughter cell will always end up with half the original complement of chromosomes. In a human, this means the original cell will have 46 chromosomes, but the daughter cell will have 23. Before meiosis can occur, the cell must replicate its DNA, just as it does in mitosis. Meiosis occurs in two stages, Meiosis I and Meiosis II. The phases of Meiosis I are Prophase I, Metaphase I, Anaphase I, and Telophase I. In Prophase I, homologous chromosomes align as sister chromatids join at a centromere. Next, they will cross over. Crossing over means that part of the DNA from the maternal chromosome will cross over to the paternal chromosome and vice versa. This recombination is a very important step in maintaining genetic diversity. The resulting chromatids now have a different genetic composition from prior to the crossover, resulting in genetic variation, and the sister chromatids are no longer identical. In Metaphase I, the meiotic spindles form and the homologous chromosomes attach to it. In Anaphase I, the homologous chromosomes are pulled apart until there is a pair of chromatids at each pole of the cell. Remember, the chromatids are still attached to each other during Meiosis I. In Telophase I, the spindle breaks down, then cytokinesis occurs. Each daughter cell will have one homologous chromosome from each pair, still with two sister chromatids. In Meiosis II, the sister chromatids will be split up such that each daughter cells will have only one set of chromosomes, that is, half the number of chromosomes of the regular body cell. In Prophase II, the meiotic spindles start to form. By Metaphase II, the meiotic spindle is completely formed and the sister chromatids line up and attach along the center of the spindle. In Anaphase II, the sister chromatids begin to pull apart and move towards the poles of the cell. Finally, in Telophase II, there are a collection of single chromosomes at each pole of the cell. In humans, there will be 23 chromosomes at each pole. A new nuclear envelope will form around these chromosomes. After this stage, the cell will divide via cytokinesis, resulting in two new daughter cells.